My name is Meyer. I'm an associate professor of bioengineering at UCLA. I also serve as the vice chair of the Department of Bioengineering. I joined UCLA in 2017 after conducting PhD work at the Massachusetts Institute of Technology, and then started my lab as a fellow at the Koch Institute for Integrative Cancer Research, where I learned a lot about using an engineering mindset to study how cells sense and respond to their environment. My research stems from a fascination about how cells communicate and coordinate their function to do useful tasks. The immune system is a perfect example of this coordination, where different cell types, such as T cells, B cells, and macrophages, work together to protect us from disease. My lab develops new ways to measure the pathways cells use for this communication, and then we make sense of these measurements with mathematical and computational models. These help us to see patterns that we would otherwise miss, and ultimately design therapies for diseases such as cancer, infectious disease, and autoimmunity. I'm especially excited by a recent discovery of ours about how cancer cells avoid the immune system. Generally, we can break up the adaptive part of our immune system, the part that can learn the difference between cancer and healthy cells, into two parts, T cells and antibodies. About 80% of cancer patients make antibodies against their cancer cells, just like your body makes antibodies against a virus when you have an infection. Our own antibodies are important because they can adapt just like cancer to prevent cancer from coming back. While patients make these anti-cancer antibodies, they aren't effective in killing cancer cells. Because they're just one protein, antibodies depend on communicating to other cells of the immune system to do useful things, like killing a disease cell. We know that part of an antibody, the Fc domain, interacts with receptors on the surface of many types of immune cells. And depending on the message this Fc domain contains, antibodies drive different types of immune responses or even prevent an immune response. Therefore, I hypothesize that the Fc domain of these antibodies is modified in such a way that prevents interaction with the immune system. And this is exactly what we found, that there's a sugar attached to the Fc domain of these antibodies, preventing their ability to direct immune cells. This finding means that by removing this sugar, you could reactivate your own antibody's ability to kill cancer cells. And we're currently testing this approach in the lab with an enzyme that could be delivered to a tumor. So far, we've examined antibodies from ovarian cancer patients and seen promising results. And we hope to expand our research into many other types of cancer and are optimistic about the potential this therapy could have. I'd like to take this opportunity to encourage folks to reach out if they'd be interested in collaborating, and I'm especially interested in examining other types of cancer where our findings could lead to new treatments. The intellectual property for enzymatically reactivating these antibodies is also available for licensing, and I'd be happy to speak with partners for further developing these approaches. Thank you. It's been a pleasure sharing my lab's work.